Welcome again, my dear students. Today we will be talking about structure of monocot embryo and then the development of monocot embryo. The monocot embryo, it possesses a single large cotyledon which is named as scutellum. This is very much important. याद रखना. Monocots are named as monocots because of this fact. They possess a single cotyledon. Okay, and that cotyledon is known as scutellum. The pilumule or embryonic shoot and radicle or embryonic root, in case of monocots, they are placed laterally. Here you can see this is the pilumule and this is the radicle. They are placed laterally and the cotyledon, which is named as scutellum, okay, it is upright, or you can say the cotyledon, the excessive growth of cotyledon, the rapid growth of cotyledon has pushed the pilumule to a lateral side. Okay, the pilumule, in case of monocot embryos, it is covered, it is protected by a covering of leaf-like structure which is named as coleoptile. Here you can see this structure, this leaf-like structure which covers the pilumule. This is known as coleoptile, and the radicle, in case of monocot embryos, is also covered by a leaf-like structure, a sheath-like structure rather, named as chlorhiza. Okay, so in addition to root cap, the monocot embryos, in addition to root cap, the monocot embryos show an extra covering of their radicle which is known as chlorhiza. Okay, so you can simply remember that the monocot embryos, they possess a single large cotyledon named as scutellum. The pilumule and radicle in these embryos, they are lateral in position. The pilumule is protected by a covering of leaf-like structure which is named as coleoptile, while as the radicle is protected by root cap and chlorhiza. So here you can see this is the seed of a monocot plant, for example maize. This region of the seed, it is constituted of the endosperm, which is the food material for the developing embryo, and this is the embryo. Okay, this embryo, if you can clearly see here, it is constituted, it is having a single large cotyledon named as scutellum. Then you have this pilumule or embryonic shoot, it is covered by this coleoptile. Then we have the embryonic root or radicle, it is covered by the chlorhiza. Okay, and here in this image you can see the cotyledon. Okay, it has grown rapidly and pushed the embryonic shoot to one side, it has pushed the pilumule to one side. Okay, I will repeat these things. The monocot embryos possess single large cotyledon named as scutellum. The embryonic shoot or the pilumule and the embryonic root or the radicle, they are placed laterally. They are both the embryonic shoot, or you can say pilumule, and the radicle, they are covered by sheath-like structures which are respectively named as coleoptile and chlorhiza. Okay, now the development of embryo, monocot embryo, or you can say embryogeny in monocots. Monocot embryos are also named as grass type of embryos because grasses are the representatives of monocots, and the embryo development in monocots has been extensively studied in grasses. Okay, so you can use the term grass type embryo. The embryo development, it takes place in a number of steps. We will try to remember all these steps one by one. The zygote or oosphere, it elongates along the micropylar chalazal axis. Just like in case of dicots, the zygote prior to divisions, it elongates along the micropylar chalazal axis. It undergoes the first transverse mitotic division. The zygote undergoes first transverse mitotic division and gives rise to terminal or apical cell, more appropriately apical cell. Okay, you can say it gives rise to apical cell towards the chalazal end, and basal cell or bottom cell or suspensor cell towards the micropyle. याद रखना, this is very much important. The basal cell or suspensor cell, it enlarges without undergoing any divisions. It enlarges and functions as a hostorium. So अगर हम देखेंगे जो मोनोकॉट्स का जो सस्पेंसर सेल होता है, it directly functions as hostorium. It does not divide. So you can say that the suspensor cell or basal cell, it has no role whatsoever in the formation of embryo in case of monocots. And alternatively, you can say that the entire embryo in case of monocots, it is derived from the apical cell. Dicots में different है. Dicots में जो suspensor cell है, that contributes, okay, although less, but it contributes to the development of embryo. But in case of monocots, the suspensor cell, it has no role in the formation of embryo. Now what is the fate of terminal cell? अब जो terminal cell है, okay, or apical cell, more specifically apical cell है, the apical cell, it divides by another transverse wall to form two cells. अब जो आपके पास apical cell थी, it divides by transverse wall and results in the formation of two cells. The top cell or the cell placed at the top, it divides repeatedly and forms pilumule and a single large cotyledon. The cotyledon, which is called as scutellum, it grows rapidly and pushes the terminal pilumule to one side. तो आपके पास अभी apical cell जो थी, that divides transversely and results in the formation of two cells. The top one, the top cell, it divides repeatedly and forms pilumule and cotyledon. Because of rapid growth of cotyledon, okay, the pilumule, it comes to lie on one side. The middle cell, after a series of successive divisions, it forms hypocotyle and radical. तो जो बीच वाली सेल होती है, it divides repeatedly and forms hypocotyle and radical. Both pilumule and radical, they get covered by sheaths which develop from the scutellum. जो pilumule होता है और radical होता है in case of monocots, they get covered by the sheaths which develop from the scutellum or cotyledon. Okay, and these sheaths are respectively named as coleoptile and chlorhiza. याद रखना, the sheath which covers the pilumule, that is named as coleoptile, and the sheath which covers the radical, that is named as chlorhiza. Now you can see the sequence of embryo development in case of monocots. First of all, you have the oosphere or zygote. It elongates and divides, resulting in the formation of basal suspensor cell, basal cell or suspensor cell, and the apical cell. Okay, the basal cell, it does not divide further. Instead, it enlarges and functions as the hostorium, while as the apical cell, it divides transversely, resulting in the formation of top cell or terminal cell and the middle cell. Okay, the top cell or terminal cell, it divides repeatedly, resulting in the formation of a large number of cells, which ultimately get converted into pilumule and cotyledon. So the pilumule and cotyledon in case of monocots, they originate from the terminal cell. Okay, the terminal cell gives rise to pilumule and cotyledon after a series of divisions, while as the middle cell, it divides repeatedly and results in the formation of the hypocotyle and the radical. जो hypocotyle है, this region which constitutes the hypocotyle, which gives rise to hypocotyle, and the radical, they originate from the derivatives of middle cell. Okay, and now the cotyledon that has started initiating here, it divides the cells of the cotyledon. They divide rapidly. Okay, this leads to rapid growth of the cotyledon, and because of this rapid growth of cotyledon, the pillimule comes to lie on one side. यहाँ पे आप देख सकते हैं, the pillimule or the future shoot, it comes to lie on one side because of the excessive growth of the cotyledon. Okay, so you can say that in case of monocots, you can simply remember some important terms. In case of monocots, okay, we have single large cotyledon that is known as scutellum, and this single large cotyledon, okay, it grows vigorously, it grows rapidly and pushes the pillimule to one side. So you have here seen how embryo development takes place in monocots. I will repeat these details once again. You have the zygote first, it divides, resulting in the formation of suspensor cell, which enlarges and forms the haustorium. Then the apical cell divides transversely, resulting in the formation of the middle cell and the terminal cell. While the terminal cell, after a series of successive divisions, it results in the formation of pillimule, future shoot, and the cotyledon. The middle cell, it divides repeatedly and results in the formation of the hypocotyle and the radicle. Okay, the cotyledon, or you can say scutellum, because of its rapid growth, it pushes the pillimule to one side, and that is why you can see in case of monocot embryos, the pillimule is positioned laterally. Okay, the pillimule as well as the radicle, they are positioned laterally. Okay, so this is all about the development of embryo in monocots. See you soon with a new lesson. Till then, Allah Hafiz.